Step 1 is initiation. This step begins upstream of the gene that needs to be transcribed. RNA polymerase binds to the promoter region of the gene, which is an area high in A's and T's. RNA polymerase moves along the DNA until it reaches the start sequence of the gene.
Step 2 is elongation. The complementary mRNA strand is assembled in the 5' to 3' direction on one side of the DNA. This chosen side of the DNA is called the template strand, while the strand that is not being transcribed is known as the coding strand. Remember, in this step, uracil complements adenine. Behind the mRNA, the DNA is rewound.
And the final step is termination. This occurs when RNA polymerase reaches the end of the gene, known as the terminator sequence. Transcription is now over and the DNA is back to its original double helix shape.
However, this mRNA molecule cannot leave the nucleus in this form. This mRNA molecule is known as the primary transcript and must undergo several modifications before leaving the nucleus. These modifications will protect the molecule from degrading in the cytoplasm. These modifications are a 5' cap at the 5' end and a poly A tail.
One more very important modification is the removal of introns, which are non-coding segments. These are removed by spliceosomes. Now the transcript only contains coding portions called exons. This is the final transcript and is not allowed to leave the nucleus.
The second stage is translation and occurs in the cytoplasm. The final transcript of mRNA travels to the ribosome, which is made of 2 subunits, large and small. The large subunit also has an A site and a P site, which stands for the acceptor site and the peptide site respectively.
The first step is initiation. Sounds familiar. The ribosome sandwiches the mRNA and then reads the mRNA codons until it finds the start codon, which we know is AUG. The start codon is in the P site of the ribosome. The tRNA molecule matches the codon with its anticodon and taxes methionine, an amino acid, to the site of initiation. The second step is elongation. This step is happening while the first step is taking place. The second codon is in the A site and the corresponding tRNA brings the next amino acid. A peptide bond is formed between the methionine and the second amino acid. Then the ribosome moves along the mRNA and the second codon moves into the P site while the third codon takes its place in the A site. The first tRNA detaches and the third tRNA brings the corresponding amino acid. This continues until a stop codon is reached.
The third and final step is termination. As soon as the ribosome reaches a stop codon, UAG, UGA, or UAA in the A site, the ribosome disassembles and releases the polypeptide and mRNA molecule. The polypeptide can then coil or fold into its 3-dimensional form.